So hi everyone. Hope I'm audible to you. So let's wait for some 5 minutes so everybody will join. Then we will start the class.
Good evening, ma'am.
Uh, Hi. Yes. Good evening. Do you want to tell something? Am I audible to you?
Yes, ma'am. Start class, ma'am. I will answer. Tell me, ma'am.
So okay. So I'm sharing the slides. So we will discuss the questions. Hello. Hi. Hello. Sir, can you keep at the reception? 263 room number. 263. Hello? सर रिसेप्शन पे रख दो.
Hola.
हेलो. हाँ सर, OTP 6598 263. Okay, so the first question is heme erythrin holds the oxygen as the option A is hydroperoxide and the option B is superoxide. So first of all, heme erythrin, you have to tell me the active site of heme erythrin, which metal is present in the heme erythrin.
Iron, ma'am.
So iron. So iron in heme erythrin binds to the oxygen in which form? Hydroperoxide or superoxide?
Superperoxide.
Superoxide.
Superoxide. So what is the
Yes, ma'am. Super.
haha? What?
Superoxide.
So superoxide. In superoxide, oxygen will exist in which form?
O2 minus.
O2 minus. Right. And in hydroperoxide, O2 minus 2. Right. So in hemerythrin, if you will see the structure of hemerythrin, if you will see, hemerythrin is a non-heme. This is a non-heme iron-based protein which is used for oxygen transfer and storage in invertebrates. So this is the this is the deoxyhemerythrin and this is the oxyhemerythrin. So if you will see the the oxygen will bind to the iron, and here HOO negative. So it is basically a hydroperoxide. So hemerythrin holds the oxygen as hydroperoxide, not in superoxide. Understood. So you were telling superoxide, but it is hydroperoxide because you see this is deoxyhemerythrin in which iron is in plus 2 oxidation state, both the iron in plus 2 oxidation state, and here both the iron is in plus 3 oxidation state. Okay, so here if you will see oxygen will come, both of the iron will give the electrons to the oxygen and it will become O2 2 negative. Okay, so it will basically exist as hydroperoxide form. Understood? So the option A is the correct answer. So hemerythrin hold the oxygen as hydroperoxide. So now the next question is also on the base of hemerythrin only. So first I will I want you to see the active site of hemerythrin carefully so that you can answer the next questions. Okay. So you see the active site. This is deoxy and this is the oxy. So you see the structure carefully so that you will be able to the you will be able to answer the next questions. Okay.
Yes, ma'am.
Okay, so we will move to the next question. So, what will be the coordination atmosphere of two iron centers in the structure of deoxyhemerythrin? So, the option A is pentacoordinate, tetracoordinate, B is pentacoordinate, pentacoordinate, C is tetracoordinate, hexacoordinate, and D is hexacoordinate, pentacoordinate. So, now just now only I showed you the structure of deoxyhemerythrin. So, what is the coordination atmosphere?
D. Hexacoordinate and pentacoordinate.
Very good, very good. So, the option D is the correct option, hexacoordinate and the pentacoordinate. So, as I showed, if you will see, this is the deoxy. So, in deoxy 1, 2, 3, 4, 5, this is hexacoordinate, and here 1, 2, 3, 4, 5, this is pentacoordinate. If they will ask about oxyhemerythrin, then what is the coordination sphere of oxyhemerythrin?
Hexacoordinate, hexacoordinate.
Yes. Then it will be hexacoordinate. Okay, nice. So we will move to the next question. So the hemerythrin is a option A is dimeric oxygen binding protein, monomeric oxygen binding protein, trimeric oxygen binding protein, and tetrameric oxygen binding protein.
Monomeric.
Oxygen bind. Yes, only one oxygen will come and bind to the hemerythrin. So it is a monomeric oxygen binding protein. The extent of pi electron conjugation in macrocyclic ring of heme, coenzyme B12 and chlorophyll follows the order. Have you understood the question?
Answer A. Answer A.
Answer A. Okay. So in uh,
Heme is a more conjugation 11.
huh?
Chlorophyll 10, I think so. Coenzyme the least one.
Okay. In coenzyme B12, which system will exist? This is the chorin system. Okay. So in heme, there is a porphyrin system which is more conjugated. Then in chlorophyll, this is chlorine system. And then in vitamin B12, this is chorin system. So porphyrin is more conjugated. After that, chlorophyll, and after that vitamin B12. So this, if this is corol, this is less conjugated. So the more conjugated is porphyrin system, which is in heme. After that chlorine, which is in chlorophyll. Then chorin, which is in vitamin B12, and the less conjugated, the least conjugated is corol. Okay, so according to the question, option A is the right option, right? 1, then 3, and then 2. So this you have to remember that porphyrin, then chlorine, then chorin, and then corol. This question I think they have asked in UGC net also, so it is good that you remember this question. Okay. So after that, we will move to the next question. What will be the O stretching frequency in oxyhemocyanin and oxyhemerythrin respectively? So tell me. Yeah. So what will be the O stretching frequency? I think according to this will be C option, C 744 and 844, because here in oxyhemocyanin it will bind in the it will exist as O single bond, O negative. It is single bond, so the stretching frequency is less. But in hemerythrin, there is some double bond character, due to which there is a large stretching frequency than that of the hemocyanin. So the option C is the right option, 744 and 844. So in biological system, the metal ion involved in the dioxygen transports besides iron is. So basically, we have seen the like metalloproteins containing iron, which is responsible for the oxygen transport. So they're asking, like in the biological system, beside iron, like expect iron, which other metal ions are involved in the oxygen transport? Magnesium,
Copper.
copper, yes. Because no magnesium, no zinc, no cobalt, so only copper is present in the biological system, which is responsible for the oxygen transport. Expect iron. So which of the following metalloprotein does not have iron in their active sites? So if you will see hemoglobin, this is purely a heme and iron based protein.
Hemocyanin.
Hemocyanin. Okay, so in hemocyanin, which other metal is present? Which metal is present in hemocyanin then? If not iron, then which metal ion?
Copper, copper.
Yeah, copper. So copper is present in the hemocyanin. So copper is the right answer.
Copper anhydrate.
Okay. So copper is present in the active site of hemocyanin. So this is the deoxyhemocyanin and this is the oxyhemocyanin. So deoxyhemocyanin, if you will see, copper is present in the +1 oxidation state. When oxygen will come and bind, the copper will be oxidized to copper +2 state. Now you see the active site of this. Then the next question will be on the based of hemocyanin. Okay, so hemocyanin contain a dinuclear copper core and bind dioxygen in the cupric state. A dinuclear copper core and binds dioxygen in the cuprous state. A mononuclear copper core and bind dioxygen in the cuprous state. First of all, these two, which option is correct?
Option A.
Option A. Yeah, we can simply eliminate these two options because there is no mononuclear copper, two copper are there. So it means option A and B we have to choose from option A and B. So in A and B, a dinuclear copper core and bind dioxygen in the cupric state. Okay. So mm, in azidomethylmethrin, the iron atoms are both pentacoordinate, hexacoordinate, tetracoordinate, tricoordinate. So this we will
Hexa.
hexacoordinate. Yes, hexacoordinate. So this we have to see the active site of this protein. So this is iron. Two iron atoms are present, and both are hexacoordinated. This I already told. What is the stretching frequency of oxygen bond in oxyhemocyanin? What? Which frequency?
So 734.
755. 744 actually, but 744 option is not there, then 755. 755.
Yes, ma'am.
755. Okay, so this was the assignment. So if you are having any doubt, you can ask me if any doubt is there. Or if you want to discuss something, you can discuss.
Like, what are the iron sulfur proteins, ma'am?
Iron sulfur proteins are basically the proteins they are redox proteins. We will tell the redox protein because in iron sulfur proteins are present in the human body for various redox activities. Like whenever there we have to, like in biological system, when electron transfer will be there from one species to another species, iron sulfur clusters will be involved. So we will call as redox proteins, iron sulfur clusters, we can tell them. And also in we can categorize into two classes. So one is the iron sulfur cluster in which inorganic sulfur is present, and one iron sulfur cluster is in which no inorganic sulfur is present. They are properly organic sulfurs like binded to the S cysteine. Then we will call when no organic sulfurs are inorganic sulfur are there, we will call it as ferredoxins. Then they are just ferredoxins. But when inorganic sulfurs are present, then we will call iron sulfur clusters as rubridoxins. So basically I think four iron clusters means, I think in your syllabus it is as far as I remember, Fe0, Fe2S2, Fe3S4, and Fe4S4, right? The last is cubane like structure. The first one is ferredoxin, in which no.
Group reduction also copper sulfur proteins, isn't it, ma'am?
Uh-huh.
Group reduction
Rubridoxin.
is a copper, sorry, iron sulfur protein.
Rubridoxin is also an iron sulfur cluster, but it is
Hmm.
different because there is no inorganic sulfur in that. And here the
Oh,
iron is coordinated to all the organic sulfurs of cysteine residues.
oh. So it is not iron sulfur protein.
No, that is iron sulfur protein only. 1 minute, I will see if some slides are there. Are you able to see the screen?
Yeah, now
So if you will,
now I can see.
so if I was talking about these four, so these all four are iron sulfur clusters only, and all four we will call as iron sulfur cluster or iron sulfur proteins, or we will call collectively as redox proteins. Okay, so if we will categorize into two classes, there is one is ferredoxin, one is rubridoxins. Okay, so here ferredoxin is this one in which iron is there, but this one iron is coordinated to all the sulfur of cysteine residues. When sulfur is from the cysteine residues, we will call it as organic sulfur. Okay, so here if you will see in the second structure, one, two, three, four, these four are organic sulfur. But if you will see the sulfide bridges, there are two sulfur. So two sulfur, these are inorganic sulfur. And also we will basically we will give name to these iron sulfur clusters on the base of number of inorganic sulfur. So it will be Fe1S0 because no inorganic sulfur is there. Here Fe2S2 because two are there. So now you tell me the name of this cluster.
Or inorganic sulfur cluster.
Huh?
Inorganic sulfur.
You have to tell me the name of this cluster. I told you the name of this Fe1S0. Here Fe2S2. And what is the name of this?
Fe, Fe2, Fe3, I think. Sorry,
Fe3.
Fe3,
Huh?
S3.
S3.
Yes.
How?
So S3.
How?
Hmm.
Then,
4.
then it is Fe3, 1, 2, 3, 4 sulfur are there. Na? 1, 2,
Oh,
3,
that is
4.
sulfur. Okay, 4.
Fe3S4. And what about this?
3, 4, 1. S4.
So this is Fe4S4. So basically these all are iron sulfur clusters. Okay, understood.
Understood that. What is the name of the third one, fourth one?
This third one that there is no name like that, but here it is cubane. Cubane. This is Fe4S4 cubane like structure.
Actinoside
First
means.
two, ma'am.
First two.
One is a ferredoxin and rubridoxin. I told.
One, this is ferredoxin. All three are rubridoxins.
Oh, 3 rubridoxin.
Okay, ma'am.
Hmm. So understood.
Yeah.
Yes, ma'am.
Okay. So any other doubt?
Ma'am, I will discuss my fifth week assignment question, ma'am.
Fifth week.
Yeah, ma'am.
Fifth week. How you will get? This is fifth week. You got fifth week assignment.
Okay, ma'am.
Now I'm asking. You got fifth week assignment. Hmm. You see. You were asking the name, na? So this is 1 minute. So in which no inorganic sulfur is there, that is rubridoxin. Okay. In which two are there, that is adrenodoxin. Tell something. I don't know. Are you hearing me or not?
Yes, ma'am.
So somebody is going to discuss one question. Discuss that question, please.
Yeah, yeah. Just I'm searching.
2 iron,
Wait.
2 sulfur, it is organic compound.
Two or I did not understand. Two iron, two sulfur.
Androdoxine.
Mm-hmm.
And 3 iron 4 sulfur complex 2.
So are you reading the question? Means so okay. Who was talking? Who was discussing the question number fifth? Like assignment number fifth. You tell me the question, please.
Binding metal for ADP is the options are Mg2 plus Na plus K plus Ba2 plus.
Um, your voice. Binding metal to ATP. You are asking.
Yes, ma'am.
Okay. So the option are.
Mg2 plus Na plus potassium plus barium 2 plus.
So binding metal to the. Uh, I think barium will not be there anyway,
Yes, ma'am.
because barium will not be there. Potassium, sodium, and magnesium, na? Magnesium is there,
Magnesium
right?
2 plus.
Huh. So I feel barium we can eliminate, but barium will not be there.
Okay, ma'am.
Same, ma'am, in assignment 4, I'm the doubt in question that is which of the following is not example for iron sulfur protein? Methane monooxygenase.
Hmm.
Methane monooxygenase is sulfur protein, iron sulfur.
1 minute. First, we will discuss. 1 minute. 1 minute, please. So, I think Bhavani, I think that magnesium plus 2 will bind to ATP to act as an active like ATP. Okay. So, magnesium plus 2 will be the right option. Okay. So, okay, magnesium plus 2. So, yeah. So, now the next question doubt is methane monooxygenase.
Yes, methane monooxygenase is iron sulfur protein or not, no?
The basically methane monooxygenase. I think the question will be like to active like to show the activity of methane oxygenase, is iron sulfur cluster is involved or not, because methane monooxygenase is not an iron sulfur cluster.
Oh,
But
yes.
in order to show the activity of methane monooxygenase, that I have to see that iron sulfur clusters are involved or not. What all other options are there?
Because yeah, rubredoxin is iron sulfur protein already we discussed, and
Yes.
risky proteins also iron sulfur
Yes,
protein.
yes.
Aconitase also iron sulfur
Iron sulfur.
protein.
Yeah. Then
So
means
methane monooxygenase
yes,
is
yes. Then
not
methane mon. Yeah, yeah, yeah. Then your
yes,
question. Then your answer is right.
yes.
Methane monooxygenase is not a iron sulfur protein. Yeah, that is true. So option A will be the right option.
Uh, and another I'm having doubt that is the ferritin only for the storage of iron or transport of iron.
No, no, no. Actually, we discussed one question earlier also, in which that question I felt that was wrong only. Ferritin is
Yeah,
ferritin is
only
involved in the storage of iron.
storage only, but both is given storage and transport.
Ha, yeah, yeah, yeah. Actually, in
That
question,
question is
yes, yes,
wrong.
yeah. Then I felt that
Yeah,
question is wrong because ferritin is
yes, correct.
involved in storage and transferrin is involved in the transportation.
Yeah, I have that same doubt I'm having. That's why I asked.
Ha,
Right,
yes,
ma'am.
ha,
Thank
yes.
you.
So, what other any other doubt? Any topic or something like that? If some doubt is there, you can ask now because we
Yeah,
have
yeah,
time.
ma'am. Yeah,
Ma'am,
Ah, yes.
heme, heme coenzyme B12 and chlorophyll in the order, ma'am.
Heme. Okay, okay. Order.
Coenzyme B12, chlorophyll.
Okay, so
One
you
greater than.
that you are asking me that I have to tell again means you did not understand, right?
I will tell the question. Repeat, ma'am.
Ha ha. Okay, okay. I am telling. 1 minute. I will share again. 1 minute. Oh, it is already on sharing, right? So this was the question, right? The extent of pi electronic conjugation in macrocyclic ring of heme, coenzyme B12 and chlorophyll. Okay, so first of all, in order to tell the right order, first we should know that which macrocyclic ring is present in the heme, in coenzyme B12 and in the chlorophyll. That's why I kept this slide because in heme porphyrin ring is present. Porphyrin ring, if you will see, it is highly conjugated. You see, double bond, single double, single double, single double, single double, single double, single, like that. There is no breakage of conjugation in this ring, right? This is highly conjugated, this is fully conjugated ring. But as you will go to the chlorine, from porphyrin to chlorine, you will see double single bond here, double bond single bond, double bond single bond. Here it should be double bond, you see, here it should be double bond, but here it is single bond. It means conjugation has broken here, right? So in
yes,
chlorine,
yes.
okay, so if from chlorine to chlorine, if we will go further, then here one already bond was broken, here means one bond is less. So also along with this, there is no bond here, also there is no bond here, no double bonds, right? So it means further decrease of pi conjugation. So if we will see, the highly conjugated system is heme, then chlorophyll, then
11.
vitamin B12. Okay, so according to this question, heme, heme, then chlorophyll, then vitamin B12. Okay, so first we will go, first is heme, then chlorophyll, and then vitamin B12. So 1,
The option is first.
the option is first, 1, 3 and 2. So this question is very important. This will also come in the your UPC exam also. You have to remember. They can also ask which system is present in the heme. Then you have to answer which system is present in the heme. Which system is present in the
Porphyrin.
heme? Porphyrin. Okay. In chlorophyll, which system is present?
Chlorine.
Chlorine. Yes.
Chlorine.
In
Then the pouring.
coenzyme B12, choline.
Pouring,
So,
pour.
okay, so here there are four questions. Which system? One, two, three, and this is the fourth question. This you should remember. This question is important for IIT, JAM, NET. This question they will generally ask. Okay.
Ma'am, can you explain the structure of cytochrome oxidase?
Cytochrome C oxidase.
The oxidase, ma'am, explain that structure, ma'am.
This you are telling. This cytochrome C. Okay, means I did not understand.
Yes,
Cytochrome
yes.
C oxidase or C means what you are asking?
Cytochrome. Cytochrome, that is protein.
Cytochrome, right? That with this iron containing protein you are asking, na?
Yes, yes, yes.
But this structure is very complex, right? Here too many side this copper three.
Yeah, yes, yes, mama.
That would
That's why that SAR is explained, but it's really it will be difficult to understand. That's why I'm
okay.
asking.
That we will keep for the next class. I will also read
Okay,
about
okay,
this,
okay.
and I will
Okay, ma'am.
read about this. Then we will discuss in the next class.
Okay, ma'am. Okay,
Because
okay.
this is, I think, this will be little complex, na? So I will
Oh, okay.
simplify according to your convenience. Then we will
Okay,
discuss.
ma'am. Okay, thank you.
Hmm. That same only, na? Heme A3 is there. Heme A is there.
Oh, yes, yes.
Okay, okay.
Two heme and two copper, I think so.
Okay, okay.
Two
Understood.
copper and there. One side, three histidine is there.
Ha.
That to the one Fe, that one axial histidine is there.
Okay, understood.
Cysteine.
We will
Yeah,
discuss.
cysteine will be present or not. I am just confused because one side three histidine,
Okay.
another side one histidine axial. Two cystidines are there. Whether cystidines
Anyway, this is
are
complex.
present or not, I am confused in that.
This
That's
this is complex only. We will discuss it in the next class.
okay.
We will simplify this and
Root
then
reduction is that okay, ma'am. Okay,
ha ha.
okay, ma'am.
Hmm.
Okay, next class we can see.
We
Ma'am, root
will.
reduction is a two, isn't it, ma'am?
Sorry.
Reduction is one. Two ion cluster then.
Two
Yes,
iron.
two ion cluster. Now only you told.
1 minute. Sorry, make 1 minute. Two iron clusters. You are telling.
Yeah, yeah, yes, yes.
So what two iron cluster means? I did not understand.
Two ion centers are there, I think so.
Two irons are there
Yes.
in cytochrome C.
No, no, then root reduction. Yeah,
Uh,
just now you shown the structure, root reduction, peridoxine. Peridoxine, only one ion is there.
1 minute. This you are telling, right?
I confused it.
Can you see here? You are seeing the screen, right?
Yes, yes, I,
So
yes.
here,
The two ion cluster is there. Yes.
so
Root reduction.
first one is rubridoxin.
Oh, first one is root reduction.
First
Okay.
one is rubridoxin.
The other one, three are peridoxine.
These three are ferredoxins.
Okay, okay, I can.
And one more thing is that one more name of second one is there. Here, if you will see, that is adrenodoxin. And in adrenodoxin, if you will see, if we will replace this two with histidines, we will call it as riskase protein.
Check. Okay, okay.
Understood?
Yes, yes.
From here, you can remember, okay?
Adenodoxin we are removing.
What?
In adenodoxin we are removing them.
No, 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 no.
Hmm.
1 minute. Don't get confused. Adrenodoxin is this one, Fe2S2.
Okay. risky. What's that?
Riske, RIE
Risky. Oh,
SKE. If you
risky.
RIESKE, when in
Risky, which is uh, this unit is histidine.
when we replace cysteine with histidine, we will call
Risky.
adrenodoxin is risque proteins.
Risky. Okay. Okay.
You see the nomenclature here. You see here two irons are there, two Fe, two inorganic sulfur, two S. Here if you will see three iron are there, three Fe, but here sulfur are 1, 2, 3, 4, then 4
Four.
S. 4 Fe, 4 S. We can also write it as Fe2S2, Fe3S4, Fe4S4.
Okay. Okay.
Ma'am, what is full form of ADP, ma'am?
ADP or ATP? If you
ADP.
if you will see, ADP is adenosine triphosphate. I will write in the message only. ADP is adenosine diphosphate. This is ADP. And if ATP is there, then adenosine triphosphate. These two are the full forms.
Yes,
Adenosine
ma'am.
diphosphate and triphosphate. Understood?
Yes, ma'am.
Yes. So these are the full forms. It is good that you people are interacting today. I am feeling that today is the TA class, because in the other classes you people will simply sit and whatever I will discuss, you will not tell anything. I will simply think like, okay, only I am talking. So it is good, you should talk, you should discuss, because this is TA class, this is for you only. It is good that you are discussing. You should discuss like that only. So any other doubts?
Synthetic model of hemoglobin. The
Hemo
oxygen pockets will be shielded by bulky groups.
hemoglobin,
Hmm.
oxygen part will be shielded.
By bulky group.
No, it is not true
Because
actually.
actually oxygen is connected in up. That is axially, isn't it, ma'am?
What?
That is the fourth. That pyridine ring is there. The oxygen is connected horizontally or vertically.
It is not
Just have
about
confusion how the oxygen is connected to the ion center.
how the oxygen is connected to.
Oxyhemoglobin.
Hmm. But this is not
That
like
porphyrin ring. That is porphyrin ring is there. Isn't ma'am?
for
That will shield the oxygen pockets.
no,
The porphyrin ring.
no. Actually, porphyrin ring is in the
Hmm.
plane. But
Yeah,
oxygen
there is
will
a
come
yes,
from the out of the plane, right? So there
yes,
is
yes,
no
yes, yes,
shielding like that.
yes,
This
yes, yes,
this
yes,
terminology, whatever you are using, that is shielding, that is not correct.
yes, yes.
Okay, so
That's
this is the heme, full heme, right?
yes,
So
yes,
this is
yes.
in the plane. So
In
oxygen,
the plane.
yeah,
Yeah.
oxygen will come from somewhere upward direction.
Ox- yeah, upper direction. So there is no shielding.
Shielding like that. Yeah. If you will see this diagram, you will understand. This is in the plane, right?
Yeah,
The
yes,
oxygen
ma'am.
will come
Yes,
from
ma'am.
somewhere here.
Yes,
Okay, so it will bind to the iron.
yes.
Ma'am,
So there is no shielding.
Yeah.
ma'am, in the synthetic
In the
oxygen binding,
in the synthetic.
In synthetic model of hemoglobin or myoglobin,
Yes.
oxygen binding pocket are generally shielded by bulky group. True or false, ma'am?
This is means like I am not sure what you people are talking about shielding in synthesis model, whatever you are discussing in synthesis model, that I can't tell because I did not read about any synthesis model. But bulk and all, because of porphyrin ring, that I am not sure about this, because as far as I understood, porphyrin ring is in the plane. One side there is histidine ring, and other side is free where oxygen can come and bind. But this I have to check, like bulkiness and all this I am not sure. Means according to me, I feel
False.
means
Ma'am,
it should be false,
okay.
but yeah, I have to check once again. I will.
Ma'am, how many histidine groups are there, ma'am, in hemoglobin? Two
In
or three?
yeah, in hemoglobin, if you will see here, this is hemoglobin. So
Yeah.
all the four coordination sites will be occupied by nitrogen of porphyrin ring. Fifth will be, this is the
Hmm.
histidine.
Histidine, yes,
Yeah,
yes.
so sixth will be vacant where oxygen will come and bind. But when oxygen will come and bind, there is a histidine in somewhere amino acid is present, which will come and stabilize oxygen in this bent state. Okay, so directly to the iron there is one histidine which is binding, but in space one more we will call as distal histine, which is this histine, whatever they have shown in red color. it will be
Oh,
it will be responsible for oxygen to stabilize in this position.
oh,
So there will be two histidine. One is proximal histidine and one is distal histidine.
oh, okay. One is 196, another one is 46, I think.
Ha 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 ha.
Yes, yes.
So one is proximal histidine and one we will call as distal histine.
Ma'am, rubidoxin is two ions cluster. True,
No,
yeah,
one,
ma'am.
one. One, one.
Rubridoxin
No,
is Fe1S0.
one, yes. One, one. That's not only she told.
False.
Yeah, it is false. That's not only she told. Only one.
And that synthetic hemoglobin, whatever question you asked, that I have to check. I am not sure about that question.
In synthetic model of hemoglobin or myoglobin, oxygen binding pocket are generally shielded by bulky group.
Maybe they are asking this question on the perspective of this, like they are shielded means they want to tell, like it is not, means many other like instead of oxygen many other things are also there na. So these bulky groups are shielding the pocket so that other ligands will not come and bind. Like oxygen can come and bind, but carbon monoxide is also there which can come and bind. So these heme based protein, whatever these are bulky proteins, they are shielding that pocket so that only oxygen can come and bind. So if this is the meaning of this question, then this is true. Means I have means in lecture what sir told that he mentioned something about this.
Repeat question, ma'am.
Means I am asking like, sir,
The synthetic models.
sir mentioned something about synthetic hemoglobin model, bulkiness and all. Okay, I will check and tell. No problem.
Ma'am,
Yes.
what next, ma'am?
What next means your doubts are over.
One more question, ma'am.
One more question is there.
Yes, ma'am.
Then ask.
Which of the following is significant biological oxidizing agent? The options are iron 3 plus, NAD plus, magnesium 2 plus, NADPH.
What?
NAD plus.
Which of the following is?
Is a significant biological oxidizing agent.
NAD plus. Answer is NAD plus.
Explain, ma'am.
Oxidizing agent. Oxidizing or oxidizing? What you are telling?
Oxidizing agent. NAD plus gets hydrogen. So it's an oxidizing agent. So NADH it will be coming as. Fe3 plus is already Fe2 plus only can be oxidized as Fe3 plus. It won't be coming. Mg2 plus, I think it is an active center, so it won't be oxidizing. NADPH already in the reduction form, so it is ruled out. So NAD plus can be converted as NADH.
Yeah, she is right only.
Okay.
Nicotinamide adenine dinucleotide.
Nicotinamide adenine dinucleotide phosphate.
So it's a redox reaction. NAD plus in the oxidized form,
NADPH in the reduced form.
reduced form.
NADPH, nitrogen adenosine diphosphate. H means, ma'am?
Hydrogen.
Yes, ma'am.
So it is in the reduction form.
So, any other doubt?
Methane monooxygenase structure I want. Methane monooxygenase structure.
Hmm. So my this is visible, right? This screen, right? This is the methane
Yes, yes, yes.
monoxygenase. It is not. This is the methane monoxygenase.
So how many is it?
Two iron in plus two
Two.
oxidation state.
Okay.
Then one iron is bonded to histidine nitrogen and glutamate oxygen.
Okay, ma'am. Two histidine, two glutamine.
This you can see, right? This one.
Yeah.
This is the structure.
Yes. Okay, ma'am.
Hmm. So any other doubts are there?
Ma'am, storage of iron in body is carried out by hemoglobin. Answer is correct, ma'am.
Um, Again, you please tell.
If iron in body is carried out,
Iron in body is carried out by.
Storage of iron is carried out by ferritin.
Storage is carried out by
Transport of oxygen is carried out by hemoglobin.
hemoglobin. Transport is ferritin. Transport of iron is transferrin and transport of oxygen is
It's
hemoglobin.
wrongly in that. Both are same.
Fine. So, any other doubts? So, any doubt is there? If not, we can close class. Reply. Okay then. Then we will meet we will meet in the next class. So if doubts are done, we will meet in the next class.
Okay.
Okay, so we will meet in the next class from Tuesday. Next Tuesday, 7 to 9, we will meet. If some doubts are there, you can ask. Otherwise, you can leave the class. We will meet in the next class. Okay, Bhavani.
Okay, thank you, ma'am. Everyone has left.